you. It's always my pleasure to highlight the real excellence of MSU's faculty, staff, and students in these events. And today I have the pleasure of introducing our research presenter, Federica Brandizzi. She is a University Distinguished Professor, MSU Research Foundation Professor, and she joined MSU in 2006 as an Associate Professor in the MSU-DOE Plant Research Lab. She currently leads research on the role of endomembranes, or membranes within the cell, in plant growth, productivity, and stress response on Earth, and as you'll learn, recently in space. Since 2020, she has also served as the Science Director of the Department of Energy, Great Lakes Bioenergy Research Center, one of our most successful research programs. Dr. Brandizzi's research has been supported by the NIH, NSF, DOE, and NSF since her arrival, as well as support from the MSU Research Foundation and the Michigan Translational Research and Commercialization Award. And she's developed an exciting research portfolio in the basic and applied sciences, and she brings in about $2 million for her lab every year. The breadth of Dr. Brandizzi's expertise is sought after by national and international funding agencies, including the National Institutes of Health, for which she served on the study section of the membrane biology and protein processing biology for a 6-year term. That's not a common role for the typical plant scientist, I might add. Dr. Brandizzi is also the senior editor of the Plant Journal, one of the leading scientific journals in the plant sciences. And as exemplified by her over 150 peer-reviewed articles since joining MSU, Dr. Brandizzi is efficient in building and directing effective and diverse research plans, identifying exciting new research frontiers, and managing very large budgets. In 2018, she was elected to the rank of Fellow by the American Association for the Advancement of Science. Dr. Brandizzi completed her PhD in cell and molecular biology at the University of Rome Tor Vergata in 1997. After completing her PhD, she conducted 2 postdoctoral research experiences at Oxford in the UK. She worked on gene regulation and endomembranes in plant cells. In 2003, before joining MSU, she accepted a prestigious Canada Research Chair to establish her first independent lab at the University of Saskatchewan in Canada. Please welcome Federica Brandizzi. Thank you.
Thank you, Dr. Gage, for the beautiful introduction, and thank you all for being here today. It's my honor to be here, and I'm really excited to show you some of the research that is ongoing in my lab. So I will start by sharing my presentation with you all. And I would like to start by saying that when I joined MSU 17 years ago, I came to the realization that I could play a role in solving one of the biggest problems that faces mankind today. And that is the problem of how to sustain our growing population with a steep rise in demand for food, energy, materials for constructions, and medicines. So what we know is that, I'd like to go to the next slide. Yes, what we know is that our population is projected to rise to 9.6 billion by 2050. And we also know, though, that plants are the primary providers for food, green energy, and sustainable biomaterials that are necessary for mankind. And as such, plants can offer realistic solution to the problems that are put at hand. But as you can see from these slides, at the current rate of production, our crops cannot fulfill the necessity of our growing population. The crop productivity is not going to be sufficient. And this is aggravated by the current climate change situation that impairs crop productivity. Now, the good news is that there are solutions, and these are offered by fundamental research in plant biology, like the one that is done at MSU, and that my lab is contributing to. So, who am I? I'm a plant cell biologist, and for the last 3 decades, I've been working on the plant secretory pathway. So the secretory pathway is a set of organelles that you see in this diagram on the left. And it's highly conserved in plants, humans, and also unicellular organisms like yeast. And the secretory pathway works as a conveyor belt to produce, synthesize, assemble, and deliver proteins, essential fats, and also sugars to the cell and to the rest of the organism. Therefore, the secretory pathway essentially builds an organism. Now, when we look at how the organelles of the secretory pathway look like in a cell, I want to show you these video clips that we took thanks to the powerful microscopes that MSU provides for researchers like me. So now we're looking at organelles that are alive inside the epidermal cells of a plant. So as you can see, there is so much life, and these organelles, the endoplasmic reticulum, the Golgi apparatus, and the vacuole, make part of this chain of organelles of the secretory pathway that I was talking to you about. So it's beautiful to see so much life, and it's also beautiful to see that when we eat our salad, this is the kind of energy and life that we're eating. So just keep this in mind, sometimes at lunch or dinner today. So now through my career, I have come to appreciate the activities of the organelles of the secretory pathway can be modulated, but can also be bettered to become more productive. If the plant secretory pathway is improved, then crops become more productive. And this is achievable if we identify and understand the key players that operate in this pathway, and then we translate our findings to crops. And this is what my lab does. So we move our discoveries from the subcellular level that I'm showing here today to the field. Now, the motivation of my research is fueled by a sense of urgency and responsibility. As a mother, I want to give my 3 children a better world than what I found and change the direction of where it is going. I want it to go in the positive direction, not negative. And I know that we are also running out of time. So I want to do this now while I'm at MSU, because thanks to MSU, I have the support of an incredible team of researchers, generous funding bodies, and the awesome MSU leadership. So I strongly believe that together at MSU, we can better this planet. Now, enough about me. Let's talk really briefly about 2-3 examples of the research that is ongoing in my lab. So, and what do we do from the bench to the field? So we look at organelle morphology. We want to understand how these secretory organelles establish and maintain their identity, but also how they work together to make sure that the plant gets the right nutrients to transform them in the biomolecules that the secretory pathway produces. Among these, there are many antimicrobial compounds, and we can understand how the secretory pathway not only synthesize them, but also deliver them outside of the cell where pathogens attack the cell and the plants need to defend itself. But also, we're studying how we can potentiate the secretory pathway to produce precursors for biofuels and bioproducts, and how the secretory pathway can help us understand humans for many of the conserved mechanisms that exist between plants and humans, including how cells respond to stress. And life here on Earth is fantastic, but we need space. We need to move to the next frontiers, and these are extracellular surfaces, and this is what my lab does. We work with NASA to understand how we can take plants to space and colonize new surfaces. Now, we work with modern organisms like Arabidopsis and Brachypodium because they're easier to work, then translate our findings to crops like sorghum, alfalfa, soybean, and also poplar. So we have a very healthy portfolio of studies and research. So just a couple of examples of what we're doing more practically. So we know that plant cells are surrounded by a very stiff cell wall that gives the plant its structure. This cell wall is made mainly of carbohydrates, and the carbohydrates are densely packed with each other. So a few years ago, we discovered that a protein from cotton that we called a CGR, called cotton Golgi related protein, could be put into Arabidopsis, our model plant species, and overproduce, so reaching limits that the cell normally does not produce at the level that the cells do not usually produce. And so what we have here is an Arabidopsis plant, a wild type, and these are transgenics that overexpress the CGR protein. And you can appreciate that these plants are bigger. This is important. It's an important achievement because we know that land is limited and we need to use land the best we can. So we may have to make plants that are bigger, but also they have better quality. And I'll get back to this in a second. So we translated our finding, our trait for biomass increase to poplar. And we found out that the stem of poplar, transgenic poplar that overexpress this protein are bigger than wild type. But we also found, and this is very important too, that by introducing this gene in plants, we can increase their digestibility. And this is important because in downstream applications, for example, in the biofuel arena, it's very important to use enzymes that can free sugars from the cell wall. And then these sugars will be fermented by microbes to produce biofuels and bioproducts. So if we make the cell wall more digestible, so we need less enzymes to convert the biomass into sugars that we can utilize. And so the trait that we put not only enlarged the cell wall to make the plants bigger, also allowed better permeability by enzymes. And this is a trait that we're trying to understand for its impact into feed for animals. If the feed is more digestible, it's likely that feedstock will be able to digest the food in their rumen and therefore benefit for their health. Now, talking about health, something, and if you had breakfast this morning with cereals, I hope you did because it's very good for you. There is a sugar that we work in our lab that is called mixed linkage glucan. This is soluble fiber, and it is produced in cereals, mainly in grasses. And we discovered in our lab that is produced by the cellulose synthase like F enzyme. And we demonstrated that this enzyme is produced in the Golgi of sorghum plants. And we thought, okay, maybe we can produce even more if we overproduce this enzyme. And maybe it was an approach that was a bit, say, naive, because when we tried, we actually found out that the plants suffer. And I can go in the scientific explanation for that, but essentially we found out that our approach, just straight overexpression, was not sufficient. And this is not only us, this is a multi-million-dollar industry, so many other scientists are on the same case. Well, we actually came out with quite an, I would say, clever approach to overexpress this protein at the time that the plant would not suffer from its overproduction. And so we were successful in producing sorghum transgenics that both in the greenhouse, but also in the field, can now overproduce this very important sugar. Now, enough about Earth, let's move to space. So we've been working for years with NASA, trying to understand how we can improve plants' life in space. This is very important. For many years, NASA and many other space agencies around the world have been working towards establishing life on the International Space Station. And efforts that we contributed to led to putting seedlings into the International Space Station. And I would say with variable success. Just to give you an example, these are our seedlings brought to space. You can see that they are transparent, sick, and not really the best plants. And this is our control on Earth. So there is still some work to do, but it is promising. We know now how plants respond to space. And this has led to the next step in our endeavor to move to space. And we were really fortunate to be given the opportunity to participate to the Artemis mission, which is the first step towards Mars. Essentially, we're moving from the International Space Station that will be soon decommissioned to the moon, and then from the moon to Mars. So if we want to colonize these very important surfaces, we need to learn how plants can survive the flight, but also can they live on these surfaces. So one way to do it is to make sure that plants have enough of the nutrients that they need for the travel. And so we've been working with NASA by using a germplasm, so seeds, they have an enhanced level of amino acids. Amino acids are the precursors for peptides and then proteins. And we used a technology that we developed here at MSU that some genotypes, some backgrounds of Arabidopsis plants have more of certain type of amino acids that stimulate growth. So we flew seeds with Artemis 1, the first of the, I believe 5 missions that will go eventually to Mars, to seeds that have enhanced levels of the amino acids that make not only plants bigger, but also more equipped with essential amino acids that will allow them to grow. And we were successful in sending seeds to space. They came back. And as you can see here, they're germinating on our media in the lab, and they look healthy. And preliminary results in our lab indicate that these plants are doing better than the control when returned to flight. And so by providing them with more nutrients, we are fortifying them to resist space travel. So with this, I would like to conclude, and I would like to thank the team that's supporting me in the lab here at the Plant Research Lab, of course, all the funding bodies, and you for your attention and support. Thank you.
Well, thank you very much. And I should note that it's auspicious that this lecture is on the eve of the 150th anniversary of our Beal Gardens and the oldest experiment on Earth. And Professor Brandizzi, I believe Professor Beal and Professor Bailey would be very proud of you and their legacy in your work. And I think we should all thank our Board of Trustees for their investments in our world and universe leading investments in plant science. And I also want to thank you very much for your urgency and the responsibility that you outlined. And on behalf of MSU and the world's children, thank you for your work. We really appreciate it.